Hi everyone, this is the last lecture to wrap up chapter 10, and this lecture should be pretty short, so just hang in there. Okay, so we finished talking about transcription, translation and DNA replication for regular cells, and so now we're going to talk about it for viruses and for bacteria. So to start off with, what is a virus? A virus is just an infectious particle, and it contains nucleic acid, so sometimes DNA, sometimes RNA, and it's always going to be wrapped by a protein coat. So pretty much it's just a piece of protein that surrounds some kind of nucleic acid, and that protein coat that surrounds it is called a capsid. And there's just different shapes of viruses. It can be kind of like this shape right here, which looks like an HIV virus. Or here we've seen bacteriophages before, and you notice the capsid's on the outside, and you have the DNA or genetic material on the inside. So viruses can only replicate inside other cells because they have such few materials. They need the host cells' ribosomes. They need the host cells' other things to create more and more of their own viruses. And so there are two different life cycles that the viruses can have in order to replicate. One of them is called the lytic cycle, and this results in the lysis or the breaking of the host cell, and a bunch of viruses are just released from inside to outside. There's also the lysogenic cycle, and this is when viral DNA replication occurs without actually destroying the host cell. The virus will insert its DNA, which becomes incorporated into the host cell's DNA, and it doesn't actually destroy the host cell. So we'll go into both of these processes in more detail. Okay, this is just a picture that depicts the two halves, the two different cycles, the lytic and the lysogenic. For the lytic cycle, what happens is the virus will sit on top of the cell, and notice this example is just with bacteriophages and bacteria, but it's the same kind of process for regular viruses and normal human cells. So the virus sits on top of the cell and injects its DNA or genetic material inside. The genetic material that it inserts then forms a small circle, its own viral little DNA loop. And immediately, this viral DNA is going to be replicated and transcribed and translated to create more virus proteins and virus DNAs. And so all those things get assembled together to form a lot of new viruses. And once the cell is full of viruses, the cell explodes, and all the viruses are released, and now you have a bunch more viruses that go on and infect more cells. So this is the lytic cycle, and it ends up killing the host cell. In the lysogenic cycle, it starts off in the same way, where you have a virus that inserts its genetic material inside of the cell. The genetic material becomes a loop. And so after it becomes a loop, instead of going on to create more viruses, the loop will actually insert itself into the normal DNA. And so this virus DNA that's inserted into the host cell's DNA is now called a prophage. So now every time the cell divides, it replicates its normal DNA, and it just assumes that the viral DNA is the same as its own, and so it will replicate the prophages with its own DNA, and every time the cell replicates, it keeps on doing this, and so you can have many, many, many daughter cells that will have this inserted prophage inside of it, which is just the virus DNA. And so it'll keep on going, and normally this doesn't kill the cell, but what happens is there can actually be an environmental trigger or stress that will have the cells switch from their normal lysogenic cycle into a lytic cycle. And so what happens is these cells can actually get the virus DNA to be removed. So it goes back here. So the virus DNA or the prophage that was inserted into the host cell's DNA will actually get excised and come off and form a little loop, and then it will go back into the lytic cycle. And that's very dangerous because now you have multiple, many copies of host cells that have been infected and now go through the lytic cycle. So both of them can be very harmful to the host cell. Lysogenic cycle isn't as harmful unless it's triggered by an environmental factor. Okay, moving on to bacteria. We've talked about how bacteria normally reproduce asexually, which just means that through binary fission, the parent cell will divide and create two identical daughter cells that are genetically the same as itself. So normally there's not really too much change in DNA other than mutations, but there are actually 3 ways that bacteria can gain new genes aside from mutations. It's called transformation, transduction, and conjugation. And we'll talk about each of these in more depth. Transformation is when the bacteria cell just takes up foreign DNA that's floating around in its environment. So these might be DNA fragments from other cells that have died, and it will just get absorbed into the cell, and then parts of it will get incorporated into its normal host DNA. And so here you can just get foreign DNA from the environment that's taken in and transformed into the normal cell. In transduction, I like to think of it as like alien abduction. I don't know, it sounds similar in my head because like bacteriophages look like aliens, but in transduction you have a virus that incorporates some of a previous cell's host DNA. So it'll take DNA from one bacterial cell, and then when it injects its own DNA into another host cell, it will also inject some of the bacteria DNA that it picked up previously when it was being infected earlier. So viruses are actually like transforming or carrying bacteria DNA from one cell to another. And so this is another way that cells will get more different kinds of DNA. And the last one, called conjugation, this is kind of the equivalent of bacteria being able to reproduce sexually. And this is when there's a transfer of DNA between 2 bacterial cells that are physically united. Usually, so they'll form this kind of sex pilus, or like a mating bridge, and it forms a little like tunnel between the 2 cells and allows DNA material to go from one to another. For this to happen, it usually needs something called an F factor. An F factor is a small little circular piece of DNA that just has genes that allows the cell to form these sex pili or mating bridges. And so it replicates its own DNA, the F factor, and it goes into the other cell. I know your textbook talks about like the 2 different ways of conjugation. You don't really need to know all the details of that. All you need to know is that conjugation is, you need to know the definition of conjugation, and you should know that it usually needs an F factor to occur. All right, the last part of chapter 10 talks about plasmids. And plasmids are just smaller circular DNA molecules that are separate from the normal bacterial chromosome. So here is just like a really long bacterial DNA, and plasmids are these small little circular chunks that are just separated. We just talked about an example of a plasmid, like the F plasmid, and that allows for conjugation to occur. But you can also have other plasmids like R plasmids. R plasmids are very dangerous to us because they carry genes that allow bacteria to resist antibiotics. And so there's these little R plasmids, and if bacteria have these R plasmids, they are usually resistant to antibiotics and can survive even though you're treated to kill the bacteria. So that wraps up chapter 10. And yeah, thanks so much for watching. Bye.